CAT. The CAT gene provides instructions for making pieces or subunits of an enzyme called catalase. 4 identical subunits, each attached or bound to an iron-containing molecule called a heme group, form the functional enzyme. Catalase is active in cells and tissues throughout the body, where it breaks down hydrogen peroxide molecules into oxygen and water. Hydrogen peroxide is also known by its chemical formula H2O2. Hydrogen peroxide is produced through chemical reactions within cells. At low levels, it is involved in several chemical signaling pathways, but at high levels it is toxic to cells. If hydrogen peroxide is not broken down by catalase, additional reactions convert it into compounds called reactive oxygen species that can damage DNA, proteins, and cell membranes. At least 13 mutations in the CAT gene have been found to cause acatalecemia, a condition characterized by very low catalase activity. Many people with acatalecemia never have any health-related problems, although the condition has occasionally been associated with open sores or ulcers inside the mouth, leading to the death of soft tissues or gangrene. Acatalecemia also appears to increase the risk of developing type 2 diabetes mellitus, the most common form of diabetes. The mutations that cause acatalecemia occur in both copies of the CAT genes in each cell, and they reduce the activity of catalase to less than 10% of normal. A shortage of this enzyme can allow hydrogen peroxide to build up to toxic levels in certain cells. For example, hydrogen peroxide produced by bacteria in the mouth may accumulate and damage soft tissues, leading to mouth ulcers and gangrene. A buildup of hydrogen peroxide may also damage beta cells of the pancreas, which release a hormone called insulin that helps control blood sugar. Malfunctioning beta cells are thought to underlie the increased risk of type 2 diabetes mellitus in people with acatalecemia. It is unclear why some people have no health problems associated with the shortage of catalase activity. CAT gene polymorphisms may also be associated with high blood pressure or hypertension, a skin condition called vitiligo, thinning of the bones or osteoporosis, and elevated levels of cholesterol and other fats, also known as lipids, in the blood, which increase the risk of heart attack and stroke. However, it is unclear how polymorphisms in the CAT gene impact catalase activity, and how changes in the activity of this enzyme might influence a person's risk of developing these diseases. A large number of genetic and lifestyle factors, many of which remain unknown, likely determine the risk of developing most common complex conditions. If you have any concerns about this gene, contact a practitioner who understands nutrigenomics and epigenetics.